everyone can hear me without the speaker here? No. No.
Take the microphone, guy.
Thank you.
And open it.
עובד?
תפתח אותו. עכשיו. תפתח אותו.
לא. Hello everyone. My name is Guy and I hope you enjoy your lunch. And I would like to welcome you here to our new facility in Masada. And I will take the time to introduce our R&D center and a bit about the Dead Sea chemotherapy and how the Dead Sea can actually facilitate the treatment of skin disorders and other inflammatory disorders. So I hope you find it enjoyable. And you can also stop me at any time to ask questions. Okay. So I'll start with a short introduction about our R&D center and about myself. As I mentioned, my name is Guy. I got my diploma in pharmacology from the Hebrew University. So most of my research is actually drug-oriented or drug-driven. And besides being an investigator in dermatology, I'm the head of the Business Collaboration and Service Unit, which provides services for the industry, for cosmetic companies, dermocosmetics companies, startup companies in the field of dermatology. And we have a lot of new and exciting models, such as a model of burning, a model of peeling, and so on that they can use. And my main research interests are, of course, dermatological or herbal medicine and the isolation of new APIs from this unique environment and plants. I study also the impact on environmental effects and air pollution on the skin. On air pollution and how it may affect the extrinsic aging process in the skin. So a lot of nice data, both for product-oriented for the cosmetic, but also basic research. And lastly, I'm currently working on investigating UVB-induced damage and how actually we can harness the endogenous protection mechanism of the skin against that deleterious action. So, me and my colleagues are a part of the Dead Sea and Arava Science Center, which is a regional R&D center located in the, I would say, 1/3 of the country. We are here in Masada. We have also a branch in Chatzeva, Elot, and Mitzpe Ramon. And we have almost 40 PhD researchers in a variety of disciplines, such as ecologists, archaeologists, agriculture, and of course biologists like myself. And of course, this is our new facility, the Dead Sea Research Institute, which was only opened 6 months ago with the driven force of the Tamar Regional Chronicle and Dov Litvinov, and also with the support of several philanthropic foundations such as ICA Foundation, the Porter Foundation, and the Ministry of Science and Technology. And I will, if you have the spare time, just go around the lab. We can show you neat stuff for biologists, of course. Anyone here is a biologist? No, only water, right? Okay. So in our branch, we have archaeologists, hydrologists, biologists, and ecologists, but all of them are working on this specific environment. So our archaeologists are working on the Engedi ruins in the old synagogue over there, dated from date for the 7th century. Our hydrologist, Kamit, which she talked about, working on synchron phenomenon and floods. We have ecologists that investigate this unique environment, and biologists that are focusing pretty much on this environment. So we have medicinal plants taken from Engedi and the surrounding area, skin pathologies, which is related to the chemotherapy and the proven effect of the Dead Sea. a microbiome, which are working also on the microbiome, the diversity of the sinkholes and sites like that, and the Shminiadis, which is a major concern over here. But our model is that because we are very separated from the larger university, we have to have an edge. And our edge is actually cross-disciplinary research. So we have the investigation of the microbiome of the skin. We take the microbiota from the sinkhole and try to produce new skincare products. We have several projects of medicinal plants taken from ecology, and I will demonstrate from archaeology, and over the skin. For instance, one of my projects is actually investigating extract taken from the plant named balm of Gilad, which probably some of you will know it as the biblical afarsemon or myrrh. It was one of the gifts, the present that Jesus was given in birth. And according to legend, King Solomon was given this unique plant by Queen Sheba. And we actually have some archaeological evidence that this story is actually true, because this is taken from the old Engedi synagogue, and it actually warns people not to reveal the secret of the village, which was how to cultivate the afarsemon and how to extract the fragrant and therapeutic potential of this plant. It actually, in the old time, was the cure for everything. If you had a disease, that's what they will take it. And a few years back, agriculture from the Kibbutz Engedi replanted these plants, and we actually found an active second metabolite that actually can attenuate skin cancer. It's published, it's on PubMed, of course, and we have an ongoing project of UVB-induced, the attenuation of UVB-induced damage by another component of this extract. And what is unique is that it actually can block UVB-induced damage for a long period of time, for 48 hours. So you can apply it topically, go to the sea, and after you return home, you'll still be protected. So it's very, very interesting. So let's get back to our main topic. What is chemotherapy? Chemotherapy is defined by the treatment of diseases in a unique and suitable climate. And the Dead Sea is known for thousands of years as a place for remedy for dermatological problems. And in the last 50 years, it actually transformed from folklore to evidence-based medicine. Currently, a lot of work was performed by us and others on skin pathologies such as psoriasis, vitiligo, atopic dermatitis, but not only skin disorders can be treated here, but also lung and joint disorders, some indication of improving in diabetes and also in osteoporosis. So, hello? Okay, you can stop me if you want. So, what is so unique in this environment? First, we have a hypersalinity terminal lake, which you probably know better than I, and a unique composition of the salt in the area. So we have more salt and different composition than any other. We also, because of its terminal lake, we have a lot of deposit of biological material from the Jordan River. So we have a lot of asparagine in the water. People don't know that. Another thing is the hot springs, which flows to the Dead Sea. And the black mud that you see everyone just applying topically is actually because of them. And also they contribute to bromide in the air. So we are more relaxed over here. And selenium in the water, which is a very important cofactor in biology. We have a high and stable temperature, This is almost the lowest as you get all year. This is the lowest. Highest is about 40 degrees Celsius. Low humidity and high atmospheric pressure. That means that the pulmonary patient can breathe better here. And we have a different UV profile because of the minus 400 below sea level and the bromide in the air that actually scatter a bit of the radiation. So before trying to investigate what contribute to the climatotherapy in the Dead Sea, let's pick one disease, psoriasis, talk about it a bit, and I'll ask you afterwards, so pay attention, what you think contributes mostly to the treatment of psoriasis. So, psoriasis is a chronic common disease affecting almost 3% of the human population, equal distributed between male and female. All races have it, and it's a complex multifactorial inflammatory skin disorders, which actually have remission period and recurrent period. So people will have a lesion, it will go away, and afterwards will be, again, after environmental factor, perhaps re-inflamed. And we don't know what causes the psoriasis exactly, but we know that genetic factors, stress and environmental factors can all cause the manifestation of the disease, which looks like silver or white scaling plaque, but also can manifestate in the fingertips, in the elbows, whole body, or just by points. It's very diverse. And if we're looking about on the epidermis, on the most outer layer of the skin, we can see that after the environmental trigger, or the stress trigger, or even bacterial infection, we have a high-grade local inflammation. And this inflammation actually causes, in a vicious cycle, the fast proliferation of the keratinocytes, which are the cells of the epidermis. So if normally the keratinocytes actually divide very slowly until they lose their nuclei and terminal and die and shed from the body, in psoriasis it is much faster, 7 times more faster, and it causes the thickening and the manifestation of the plaque. So, from all of this, what do you think is contribute mostly for the successful treatment of psoriasis? What? The soup. The soup. The soup. From the, let's see, yeah, okay. What else?
I can, if you go back to one slide, I will explain it. 20 years ago, I come to work in Brom factory to start a raw plant, and I had the psoriasis. I didn't know that here in this area you can improve it. I didn't understand it. I was under very high stress because of the commissioning.
Yeah.
But after I finished the work, it is gone. I don't know why it is gone, and it's 20 years not anymore.
Okay, so that's stress.
But stress, take it out, because I was under very high stress.
Okay. So what actually can cure from here, just one going to the Dead Sea, what can actually cure or ameliorate psoriasis? Yes, it's over here. UV, that's a good guess. Anyone else?
Some of it.
The sum of it, of course, that's the Jewish answer actually. Everything matters. Yeah, so surprisingly, the UV profile is the most prominent factor from all of these. And how does it work? So first of all, ultraviolet radiation is actually divided by 3. UVC, which is very high energy, but actually blocked by the ozone layer. UVB, which is with a lot of energy, but can only permeate the skin until the epidermis, and UVA that can go further down, even up to the full dermis, but with low energy, so low effect. And UV can induce cellular damage such as DNA damage, apoptosis of the keratinocyte, reactive oxygen species. We can also see overall tissue damage because we have low collagen content because of UV and premature aging, sunburn cells, and the modulation of the immune system. So this is actually an important point. So all of these typically can cause, if we are exposed chronically, to premature aging, wrinkle formation, and also cancer formation. And if we look about the impact of the different UV length on the skin, we can actually see a very interesting phenomenon. You see, in the UVB region is more dangerous to us than the UVA. And if we look at DNA damage, it actually peaks over the UVB region and actually goes down along as we go to the UVA region. The same for erythema, and skin cancer peaks and also go down almost completely in the UVA region. And the Dead Sea UV profile is actually reversed. So we have a blockage because, again, the -70 and the bromide in the air, scattering of the UVB almost 30% lower than Jerusalem or other area located closely. And we have a lot of UVA. And this actually can modulate the immune system. So we have no adverse effect or low adverse effect with the potential of treatment. So how is it so effective? So from clinical data, mainly performed by my colleague Dr. Harari in Bokek, which is also a part of our R&D center. He is working, I think, almost 30 years over there with thousands of patients. And published data show that almost 80% of psoriatic patients have a remission or a very significant improvement, almost 75% after 4 weeks just being here in the Dead Sea. And the average duration of remission is tremendous in comparison to other treatments. So 33 weeks without the recurrence of new lesions. And we know that if we compare it to no band UV or UVA that is used in clinic, which is actually mimicking the Dead Sea, it actually looks better than them even. And of course, from a pharmacology point of view, it's much safer than drugs, which are typically very restricted steroids treatments and the new biological treatments, which are anti-TNF and anti-IL-70 antibodies. So, how do you treat here psoriasis? First of all, one should be advised to use medical supervision because the UV profile is actually changing all day and all seasons. So, and it's also actually very important to adapt the protocol to the skin type and condition. So, if I will go to him, he will say, "So you're a bit black. You can be more in the sun than one of most of you." Okay, and so another very important point is that you need to be here at least 3 to 4 weeks so you have the full beneficial effect. So typically, Israeli comes to here for 3 or 4 days over the weekend. They just don't understand why doesn't work and why they suffer for sunburns. But if you wish to do the right treatment, 3 to 4 weeks, and we have actually a lot of patients going from Germany, from Oslo, from the Netherlands, and from all the Europe, just going to over here. It is subsidized by their clinics, and they actually have massive improvement from 100% lesional to none. So I talked about the radiation, but it's not all the story, because we know that if we expose only to the sun two times a day, we have a improvement of 72%. But only bathing with in the Dead Sea, we can actually have a nice 30% improvement. When combined, we have approximately 85% success and a longer remission time. And it is actually also working when we just take the Dead Sea salt, applied it to the skin, and use the traditional phototherapy with the UV lamp that I mentioned. So we have two major factors, but the sun is the trick here. So how does it work? Again, UVR, we know that it actually attenuates the hyper-inflammatory reaction in the lesion. And we know that directly it can also reduce the proliferation rate of the keratinocytes. Also, we know that minerals such as bromide and magnesium can inhibit the epidermal proliferation rate. Again, reduction of the lesional area. My thoughts is actually that tranquility. You were working here, but if you're going to the Dead Sea for 3 to 4 weeks, just without work, just to have fun, you can't really say that tranquility doesn't provide you a lot of the effect. It's not all, but it's actually, I think, a very major treatment here. And of course, you are not in your environment. So if you had something that you react to, such as infection or dust or something, that change going over here will also contribute. So again, the Jewish answer, everything is important, but if you remember one thing, the UV profile is the most important. So beside clinical outputs, a lot of study conducted here saw gene regulation by halotherapy, dipping in the Dead Sea, and phototherapy. Of course, inflammatory marker, we saw a reduction in TNF-alpha, actually attenuating to its basal level, a reduction in IL-17. This is still unpublished, but I can share it also. And a lot of increase in vitamin D, which actually correlates to the success rate of this treatment. And if one of you is interested in this field, another publication from our organization, from our R&D center, is the importance of microbiome and microbiome on the successful rates of the treatment. And we don't know if they are the cause of one of the cause for the better treatment, or actually just modulate because of it. But absolutely, they are changing over there quite tremendously. So we talked about psoriasis, on left, atopic dermatitis, which is another inflammatory skin disorder, different mechanism, different cytokines, a Th1 versus Th2, and etc., but similar improvements. Almost 75% of the patients here, and we're talking about 1,000 a year, actually have a significant improvement in atopic. It actually almost cured them. And vitiligo, which is a depigmentation disease, also good results over here. Of course, this is probably because we can get more sun without the adverse effect of or risk of skin cancer. So they can stay here for a lot more time and try to repigment the bleach area. So the take-home message for this section is one last one. This section is we actually transform the folklore or the hypothesis that chemotherapy in Dead Sea is working to an evidence-based medicine with treatment regimes and standardized procedure, and it's actually recommended for psoriasis patients. It's actually it's the first line of treatment for all the physicians in Israel. Just sending the patient over here, and only afterwards, if it doesn't work, try steroids, try oral drugs or biologics. So transforming all that data from clinic to bench, this is our work. And we are working a primary model system. Of course, we have cell culture and everything, is the human skin organ culture. And I have to just something that you can move around. So our model system, we don't use animals. It doesn't work with cosmetics, and it doesn't really work at all because human skin and rodent skin are not quite alike. The only best, most suitable afterwards, after the human skin, is actually porcine, which have very hard to get. So we take human skin taken from abdominal surgery, cosmetic surgery, clean it up, cultivate it in an air liquid interface, and you can see the plate over when you get it. The epidermis is it's like this. The body, the epidermis is with air, and the dermis is submerged with culture medium. And we can use it to topically apply material or systematically apply materials, just if you want to use it topically or systemically in the body. And we have a lot of interesting model systems. One of them that is related to atopic dermatitis and psoriasis is actually the induction of inflammation. So this is a viable organ culture. MTT results, breathing can react to stimuli. If we expose it to UV, we have UVB-induced damage, apoptosis and DNA damage as normal skin. And we can screen for efficacy. For instance, if we have a project of medicinal plants, this can be used as a screening tool of topically applied compounds. So just an example for some of our model systems, we have a lot of data for basic research, but also a lot of data that was used by companies. As I've mentioned, we are service providers also. And we have several inflammatory measures. One of them is SDS, which is an irritant-induced skin inflammation, which actually reflects contact dermatitis. Skin inflammation used by the lipopolysaccharide, which is a general inflammation without any characteristics between Th1 and Th2 pathway, but also more targeted approach for psoriasis, such as TNF-alpha induced damage and IL-17. And for instance, you can see the skin that when we applied LPS on it, a massive increase in TNF-alpha, which is a pro-inflammatory cytokine, is gained. And the vehicle doesn't do nothing. And the steroid dexamethasone, which are used in the clinic, actually can attenuate this effect. Another example, as I mentioned, psoriasis and several other skin disorders are characterized by increase in hyperproliferation. And you can see that when we stimulate the organ culture, remove the epidermis, just test the epidermis, we can see a massive 50% increase in the BRDU incorporation, which is a marker for turnover rate, and it actually can be attenuated again by dexamethasone. And lastly, we can also monitor, because it's an organ culture, morphological changes after histology. And you can see the massive increase in the keratinocyte layer under stimulation, and the restoration of normal layer after the treatment of dexamethasone. So, I hope you enjoyed the lecture, not water-oriented. I wrote my details, but you can contact me if you just want to have a collaboration, even in water in the Dead Sea. I can address you to my colleagues, to the hydrologist, or ecologist, or whatever. And that's it. Thank you.
Everyone saw the skin?
How long can you search?
We typically maintain it up to 3 weeks with no reduction in variability in stimulation. But people actually use it. I saw an article, I think 2 years ago, for 3 months. So a long period of time. Of course, the more time you cultivate it, less effective stimulation would be on it.